Okay, this is the last recording for chapter 17, and I do want to go back. I do want to review the whole process. So, um, basically, transcription and translation are the processes where a piece of DNA called a gene that codes for a particular protein or polypeptide, that piece of DNA is called the DNA template. Template is used to make a messenger RNA molecule, which is then processed and changed a little bit, modified. The introns are removed and the exons are spliced together. There is a poly A tail added and then a guanine cap added. And the messenger RNA, the final messenger RNA molecule, leaves the nucleus, and now the next process is called translation. That messenger RNA molecule will find a ribosome and attach to the ribosome, and then the process of translation will occur, and translation uses the transfer RNA, which has an amino acid on one end and an anticodon for the complementary messenger RNA codon on the other end. So the process of translation happens in the cytoplasm on a ribosome, and transcription happens in the nucleus. And the start codon is always AUG, and it always codes for methionine. So methionine is always the first amino acid in a protein. So now we're going to learn about mutations and how mutations will affect the protein that's produced. So a mutation is just simply a change in the DNA, change in the genetic information of a cell, so a change in the DNA code. A mutation is not always bad, it's not always good, and doesn't always even mean that the protein is any different, because the way that the DNA code is designed, sometimes you can have mutations or mistakes during DNA replication that don't actually cause any problems. So one type of mutation is called a point mutation. That means just one nucleotide is different. It's just a change in one nucleotide. Point mutation at one point in the DNA code. All right, so one type of genetic disorder is sickle cell disease. It is a recessive trait, so both parents have to give their child the gene for sickle cell in order for their child to have sickle cell. The sickle cell is caused by a point mutation for one of the protein units that forms hemoglobin on the red blood cells. So there is the wild type, or really it's wild type means normal. There's the normal. I think they just don't want to use the word normal because then it sounds like if you have sickle cell, you're not normal, But the wild type is the regular copy of the beta globin protein. And so it gives you nice, round, healthy red blood cells when you have that beta globin, that wild type beta globin portion of the hemoglobin. But if you have the sickle cell beta globin, then that causes, if you have the sickle cell beta globin on the hemoglobin molecule, that causes the red blood cells to be sickle shaped, and it causes many, many problems. Blood clotting is just one. And anemia is another, so low oxygen levels. But the only difference is, you see the original DNA is CTCGAG. So during replication, there was a mistake, and there was a substitution of that original T in the top strand became an A. So it was just a mistake that didn't get corrected during DNA replication. During the proofreading or the error, I'm trying to think of the word. It's proofreading, and then there's a process after that. I can't think of the word, I'm sorry. But anyway, a mutation happened. So where there was supposed to be a T, now there's an A. So when the messenger RNA is produced, instead of being GAG, it's now going to be GUG. Okay? So GAG codes for glutamate, I believe. Glutamate, GLU, is the amino acid. And GUG codes for the amino acid valine. And there's that one difference in the amino acid in that hemoglobin protein. And it's a huge protein. It has like hundreds of thousands of amino acids in it. But that one amino acid difference causes one of the 4 proteins, polypeptides, that form hemoglobin to be misshapen, and it causes the entire red blood cell to have a different shape. Okay, so a point mutation can be a substitution. Point mutations can be substitutions, or they can be insertions or deletions. And a substitution is not as bad as an insertion or deletion, because if you insert an extra amino acid, you're going to change the sequence from that point on. If you delete an amino acid, you're going to change the codons from that point on. So a substitution mutation is not as bad, is not as serious. And sometimes it's even, so if you have a substitution, okay, sometimes it can even be a silent mutation. Because what if, remember how you have different codons for the same amino acid? There's some amino acids that have 6 different codons. So what if we come back up here and the GAG and the GUG coded for the same thing? They don't, but what if they did? That would be what we call a silent mutation. So it's really not it doesn't lead to a mutation at all. If it's a missense mutation, it's still going to code for an amino acid, but not the correct one. So the mutation that causes sickle cell is a missense mutation. Then you can have nonsense, and that's really, really bad. That converts the amino acid not into another amino acid, but into a stop codon. And it almost always means that the protein is non-functional. So we're looking at a substitution example that is a silent mutation because the original code, the original template strand had a G, and an A is substituted instead of the G. And so your messenger RNA codon ends up being GGU instead of what it would have been would be GGC. And if you look on the codon chart, GGC and GGU both code for glycine, GLY glycine. All right, so that is a silent mutation. Then we can have a missense, like we already saw one of these. And that is like with the sickle cell. Then we can have a nonsense. So here the mutation, wait, do we have a nonsense? We have a missense, we don't have a nonsense. If instead of, okay, a nonsense would be if instead of glycine, the mutation coded for a stop codon, that is called a nonsense. And a nonsense usually results in a non-functional protein. All right, now we have an insertion. So the insertion is going to change the DNA sequence from that point on. It's going to be different all the way down. So, and here's an example of a nonsense. Sorry, because the insertion was an extra adenine inserted here between the C and the T. There's an extra A inserted. And so now you have to start from that point and read the codons differently. So what we have now is a stop codon. And so all of these amino acids will not be coded for. So the protein will be missing 3 amino acids. Insertions and deletions are what we call frame shift mutations. They will change the codons from that point on. They are frame shift mutations. and they are normally more serious than a substitution. Here's an animation on the mutation types, and it's slide number 93. All right, there are some chemicals or physical or chemical agents that can cause mutations, and they're called mutagens. Like ultraviolet light is a mutagen that can cause mutations in the DNA in some of our skin cells and lead to cancer. So if it's a mutagen that is also a carcinogen, most carcinogens are mutagens, and most mutagens are carcinogenic. All right, so carcinogens are cancer-causing. Mutagens are just agents, physical or chemical substances that can cause mutations. If that mutation leads to cancer, then the mutagen is a carcinogen. So what we're saying is that most of the time, if it's a mutagen, it is a carcinogen. It is cancer-causing. And most of the time, if it's a carcinogen, it causes cancer by causing a mutation in the DNA. There is a gene editing technology called CRISPR-Cas9, and it is just used to edit DNA. It's used in basically gene therapy. So it basically can correct a mutation, okay? There's a whole explanation here, but it's just, and you can read it later, but right now I just want you to know that this technology edits the defective gene and corrects it. That's CRISPR-Cas9. And there is a figure, there's hopefully an animation. I guess not. Okay, so we have not been able to actually correct or cure a disease with CRISPR-Cas9 yet, but we are working on it, okay? So a gene, going back to the definition of a gene is a piece of DNA, a region of DNA, that can be used or expressed to produce a final product that is either a polypeptide or it can be an RNA molecule, which we haven't talked about that, but it can be an RNA molecule, polypeptide or an RNA molecule. And here is just a skills exercise, which it says interpreting a sequence logo. I have no idea, sorry. Are insulin mutations the cause of infant diabetes? And this is one that you could do. Wild type DNA, which is normal DNA, versus DNA from patient 1, patient 2, and patient 3. And then you just compare them. They're the same. Do we see any mutations? Not yet. Not yet. I don't see any mutations at all. So I don't know what to tell you really about this. Unless I just missed it, unless my eyes just missed it. Oh, okay. So right here, the wild type cDNA is just a type of DNA. It's just the DNA that's used as a comparison here. All right, so the wild type has TTC in this area. So this triplet code is, or this triplet group of nucleotides is TTC. In patient 1, it's TGC. In patient 2, it's TCC. and patient 3 is just like the original, and wild type means normal. So patient 3 would definitely not have diabetes, but apparently patients 1 and 2 could get diabetes because of just that one change in the DNA code. So it's just a single substitution point mutation. So it's just going to change one amino acid. In fact, you know what? We've got time. Okay, so let's look at patient 1 and patient 2. Okay, so what would this actually mean? All right, TGC is the DNA. So what would the messenger RNA be? Messenger RNA would be ACG. The messenger RNA codon would be ACG. Let's find out what amino acid that is. ACG. ACG. I'm looking for the codon chart. ACG. Oh, gosh. Okay, I'm just going to scroll through this way. So we're looking for the codon chart to tell us what ACG, what amino acid ACG. All right, here we go. ACG is going to be threonine. Now, remember that. Threonine is for patient number 1. Now let's go back to, and this is slide, I'm writing this down, slide 23, so I can come back to it. Now we're going to go to patient number 2 and see what the messenger RNA codon would be. All right. All right, so patient number 1, we said it was threonine. Patient number 2, the DNA is TCC. So the messenger RNA codon would be AGG. Now let's go back up here to our codon chart and find AC. What did I just say? ACG. Oh my God. All right, let's try this again. I just forgot it that quick. All right. All right. Patient number 2 would be AGG. AGG. All right, AGG on the codon chart is arginine. All right, so it is a different amino acid. Patient 1, it coded for threonine, and patient 2, it coded for arginine. So it is just that one amino acid difference that is causing the diabetes. In fact, so we got threonine in patient 1, arginine in patient 2. What would the patient 3, who's normal, what would patient 3 be? Patient 3 who didn't have a change. That one is TTC, which would be AAG, all right, AAG, right here, is lysine. So the original DNA sample codes that particular codon codes for lysine. Then patient 1, it codes for threonine, and patient 2, it codes for arginine. So one of those causes infant diabetes. So anyway, that's just an example of how you can use information like this to find out, like, because we can sequence everyone's DNA now. We can do DNA tests and sequence we can do it in a science lab, in a crime lab. You can send your DNA off for paternity testing, or you can send it off to ancestry.com. You know, there's all these different ways that you can get your DNA sequenced, your DNA code, and determine, a lot about your genetics, So if we can do that, we can also figure out like which difference is causing a disease. So that's what this example kind of shows us. All right, that's it. This is the last recording for chapter 17.